So welcome, everyone. I'd like to welcome you to our, I believe it's 9th or 10th edition of the Proteoform Thursdays. And today we're fortunate to have Dr. Corinne Lutomski with us from Oxford to speak about some of her recent work with the native top-down mass spec. So it should be pretty fascinating things to hear about. Let me just give you a quick update on what the consortium has been up to. Basically, there's two main areas, a lot of you know. One is we work around trying to build the community through events such as this, the educational things. We have a short course coming up next year at ASMS, and trying to build some bridges between the different parts of the organizations that were involved. There's lots of vendors that create technology. There's research institutions like a lot of you are involved with, and there's people who use the information like a lot of the biofarmers. So we're trying to knit the community together so that we can actually address the important issues. And the way we often do that is developing protocols and methodologies, creating standards, and through a variety of initiatives. Some of the ongoing initiatives, we may have mentioned these in the past, just want to give you a quick update. Frederick Lermite's leading our native top-down project. We're trying to assess the situation out there and come up with some basic guidance for people who are looking for an on-ramp to do native work. And we know it's somewhat challenging, but the groups that do it are outstanding, but the groups that want to get into it, there's some challenges. So we're trying to address some of those challenges and give people some assistance. A second is the pro forma notation, which is something we published a couple of years ago, but in collaboration with the HUPO PSI group led by Juan Vizcaino, we're updating that to include shorter proteoforms, also known as peptides, as well as some other use cases, so that we'll have this kind of universal language that's both human readable and machine readable. And that'll be something that also facilitate data sharing as well. We have a CEMS initiative. Again, CEMS has great potential, and there's been some awesome publications around some of the protein separation work. But again, there's challenges for people who are trying to attempt this work. So Liang Sun, Alexander Ivanov, and Kevin Jost are leading this work where they're assessing the state of the art through a variety of laboratories, doing some standard work to cover a lot of the different platforms that are out there, try to establish some guidelines around how you go ahead and move forward with this. We have our Human Proteoform Atlas work led by Hartmut Schluter and Neil Kelleher, where we're trying to build up the basic structure and the platforms for hosting all the human proteoform information and trying to drive that forward. This is sort of a subset of the real main thrust of our work over the next few years, which is the Human Proteoform Project, which is a paper we published last month in Science Advances. It's open access, and it's been downloaded almost 8,000 times. So if you haven't had a look, you can go ahead and take a look at that. But basically, we're proposing this ambitious project to really explain the chemical diversity of the proteoforms that are expressed from all these genes and these gene products. And along with the information on the proteoforms, we think this will also drive a lot of technology of evolution as well. A lot like happened for the Genome Project. So we're pretty excited about this, and we'll be having a variety of activities centered around this to get the community engaged, get your input on how we move forward, what are the kind of steps and milestones we want to put in place for this. And this will be something involving all of us to try to move this forward, as well as trying to stimulate more funding for this arena, which clearly would benefit from it. So if you're not a member yet, feel free to join us. Currently free on the website. You can go there and click and join up. And thank you primarily to our major sponsors, Thermo Fisher, Brooker, and SciEx, as well as our supporters, Agilent and Pfizer, who help us do these types of things, enable us to carry this work forward. So if there are folks who would like to get involved on the sponsorship level, please contact me, paul.danis@tdpro.org, and we'd be happy to speak with you about any potential arrangements where we can provide you with contact with the community, and you can help us go forward with these types of activities. So with that, I'd like to turn it over to Dr. Corinne Lutomski. She's a senior researcher and co-principal investigator at the Kavli Institute for Nanoscience Discovery at the University of Oxford. And she'll be speaking with us today about connecting proteoform diversity to biased signaling using native top-down mass spectrometry of G protein coupled receptors. So go ahead, Corinne, it's all yours.
Great, thank you. Okay, we're all set up. Yeah, so thanks for inviting me. I'm happy to be here to tell you a little bit about some of the work that we're doing here. So my talk today is on the end goal is connecting the proteoform diversity in GPCRs to their bias signaling output. And the way that we're going to do this is using native top-down mass spectrometry, and I'm going to kind of walk you through the story as to how we're going through developing that and how that's our end goal. And so this is a collaborative effort. It's a pretty major effort between the Oxford team. So in Carol Robinson's lab, that's Tarek and I, Iddy at OMass here in Oxford, and then the guys at Thermo Fisher. We've got John and Josh that we work with every week, and Chris Mullen as well. And so I do want to acknowledge, again, I just briefly introduced the team, but this has been quite a remarkable effort. So we're Oxford based, we're in the UK, and we started this project around October 2020, and so well into the first lockdown in the UK. And there's been 3 lockdowns since. So traveling to San Jose has been, not exactly all that feasible. And so we had to get creative. And so what we actually do is the Oxford team, which is Tarek and Iddy and I, we make all the proteins, we gather all the systems, we do everything we can, all the preliminary experiments on the Oxford side. And then we ship those samples all the way to San Jose, as quickly as possible. And so those samples make this 5,300-mile trek. And so every Tuesday night, even though there's an 8-hour time difference, it's our Tuesday night, it's Thermo's Tuesday morning. We meet and we run experiments remotely. And so it's actually been, it's been a lot of fun and something I look forward to every Tuesday. And so this is basically the culmination of some of that work that I'll talk to you about. So my focus is on protein receptors. Protein receptors essentially dictate how we interpret the outside world. So this is just a picture of receptors that would exist in someone's brain. But within your body, you have peptides, hormones, metabolites, even drugs circulating, and receptors are the ones that pick that up. And so when a receptor picks up a ligand, a drug, or a peptide of some sort, it then signals an intracellular response. And so that protein receptor will then interact with secondary messengers on the inside of the cell, move on to other effectors, things that affect other proteins, such as ion channels, transporters, what have you. And it's really these protein networks that initiate an output. And these protein networks, in kind of an abstract level, it's these protein receptors and these networks that really give us the human experience. And so what we'd like to do is we'd like to use native mass spectrometry, in particular with top-down native mass spectrometry, to be able to pull these receptors, these complex receptors, and complex with their ligands or other interactors, directly from human tissues, and be able to interrogate these things intact in the mass spectrometer, but then dissect them into their constituent parts. And so that way we can start to look at these proteoform specific pathways that delineate not only just healthy versus dysfunction, but again, just the differences in phenotype and physiology and the human experience. And so in our top-down workflow, we will take the native protein complex, we can measure it by mass spectrometry, we can then select and activate it. We can then get the protein or the proteoform, any lipids that would bind, any ligands that would interact, and then through multiple stages of tandem mass spectrometry, continue to dissect those individual components. And if we do this enough times, and if we have enough samples, we can start to parse out interaction networks or prerequisites for healthy receptor function versus a case where someone has, a dysfunctional receptor that would create, some undesirable effects. And so my work focuses primarily on G protein coupled receptors, or GPCRs. If you opened a biochemistry textbook, this is essentially the schematic that you would see. GPCRs are characterized by these 7 transmembrane domains. They have these extracellular bits. The loops are where the ligands will come in and interact. When you have an extracellular ligand binding to this protein, the intracellular, the cytosolic part, will interact with G proteins, which then go off and interact with effector proteins, second messengers, later effectors, and ultimately give you some intracellular response. And so there's a lot of interest and a lot of work that's been done on these protein networks, so things that GPCRs interact with downstream, and it's quite well characterized, what types of protein networks will give you a certain response. But something that's a little bit less characterized is the GPCR itself, or the proteoforms that contribute to that. And so this is just now a cartoon of you may have a GPCR. These get trafficked to the plasma membrane for the most part, so they'll have glycosites on the N-terminus. But we have, in this cartoon, we have this proteoform of a GPCR. Ligand A will come in, it will bind to the protein, and then on the inside of the cell, you have some intracellular response A. You may have the scenario, and this is actually quite a common scenario, where you have differences in proteoforms. So even the addition of another glycosylation state can inhibit now ligand A from binding, but you can now preferentially bind, say, ligand B. And so in doing so, you now have a completely different intracellular response that interacts with other effector proteins or different proteins downstream. And so it's really this GPCR proteoform diversity that not only influences ligand engagement, but the entire intracellular response and the entire downstream response. And so we do want to directly interrogate these membrane protein ligand complexes. So which proteoform is interacting specifically with which ligand. We then want to be able to select the individual subspecies and do top-down mass spectrometry to get true molecular identification of what these complexes are. So our solution, we use the Thermo Orbitrap Eclipse. This is a great instrument. It meets our requisites for our experiments. And the two major that I just talked about was that we first want to directly interrogate membrane proteins and their associated complexes. And then two, we want to select ions for top-down sequencing, and the Eclipse allows us to select in both the quadrupole or the ion trap. But I do want to go back to this first point, being able to directly interrogate membrane proteins and their associated complexes. This one is actually kind of a loaded requisite because membrane proteins are hydrophobic. They exist in your membranes. And so in order to extract these things, you do need to extract them in either detergent or some form of membrane mimetic to keep them stable. And so our instrumentation and our experiments require us to be able to then liberate the membrane proteins from the detergent or the associated mimetic. And most of these detergents are not quite mass spec friendly. And so we need to do this in a very robust way. And so on the Orbitrap Eclipse, we can actually, we can activate in source. And so I've now shown you a little cartoon or I'll walk you through a cartoon as to what this looks like. So if we apply no energy in the source, if we just, electrospray these membrane protein complexes in detergent, we generate a spectrum that looks like this. And it looks quite nice and clean. But unfortunately, these evenly spaced singly charged peaks indicate that it's, these are just detergent clusters that we're seeing. It's not our protein of interest. And so if we just continue to up the collision energy just a little bit, the spectrum gets a little bit uglier, but we start to see some semblance of a protein distribution coming out. And then we can continue to step through different collision energies until we've then liberated the protein from the detergent micelle. We can now generate these very nice spectra of natively folded membrane proteins, and in this case of natively folded GPCRs. Our second requisite is to be able to select ions for top-down sequencing. And so this was actually already demonstrated in a paper that came out from our group just last year. And so this is an example of an outer membrane protein complex, OMP-F. And so this is OMP-F bound to lipids, peptides, and drugs. And you can use this multi-stage tandem mass spectrometry approach to then select ions of interest, fragment them, and then continuously break apart these interactors, these ligand interactors. And so this worked quite nicely for a while. It still does, but there's always room for improvement. And so this is where the newest endeavor comes in. With these collision-based techniques, labile ligands tend to be ejected when we try to remove the micelle, especially with GPCRs. We tend to lose very, very fragile interactions. And so we do need a better way to liberate the protein from the micelle, but also maintain those fragile interactors. And so with that also comes the need for better fragmentation. And so depending on the detergent you use, some of them are quite sticky and not the most mass spec compatible. And so you have to impart quite a bit of energy to get that protein out. And in doing so, the consequence is that you lose signal and signal that you would need to fragment that protein. And so one of the goals that we had was to improve on some of these aspects of membrane protein native mass spec. And so the improvement that was made is we fitted an IR laser to the back end of the Orbitrap Eclipse. This laser gets fed into the ion trap. And so there's a few advantages that I've outlined. Not only do we expect to get improved sequence coverage because we'll have higher signal, we actually have more tunable parameters compared to the collision-based desolvation. And so what I mean by that is, the collision energy, you can just ramp the energy. But with the IR laser, we can tune the laser power and the amount of time that the ions will see that laser. And so we do expect that, without undergoing hundreds of thousands of collisions, that we can expect to preserve some of these fragile protein ligand interactions. And so just a cartoon, another cute cartoon to kind of highlight, how we're expecting this desolvation or this, membrane protein liberation to work. We have our membrane protein that's encased in this detergent micelle, and we're going to irradiate it with the laser for some amount of time. And so rather than the membrane or the detergent kind of, breaking off or colliding off this protein, we're now it's like a soft boiling off of the detergent over time. And then we're ultimately left with the bare protein. And so what this looks like in practice. And so we now have, this is a GPCR that's encased in DDM detergent. We can vary the laser power versus the irradiation time. And so this is an example of a relatively low laser power. This is 8%. At only 5 milliseconds of exposure, we again generate this very clean but evenly spaced mass spectrum that's characteristic of detergent clusters. Even at 10 milliseconds, we still have quite a few detergent clusters that are, inundating the signal. But if we irradiate for now 200 milliseconds, we can liberate quite nicely this charge state distribution now for our GPCR. We can then up the laser power just a little bit more to 12%. And now at this higher laser power, we can see that as early as 5 milliseconds, we can see some protein peaks. At 10 milliseconds, we can generate a very, very beautiful native mass spectrum of our GPCR. And again, at 200 milliseconds, we still see the same distribution. But at 14%, so if you go just a little bit higher, again, 5 milliseconds at 10 milliseconds, everything looks great. But if you continue to irradiate this protein, you essentially cook your sample. And so what we learned from this is that, there's a lot of leeway and a lot of wiggle room that we have now to optimize the conditions at which we can get our membrane proteins out of the detergent micelles. And this is huge now because, sky's the limit as to the downstream applications or the downstream tandem mass spectrometry that we can actually perform on these things. And so the first example that we used to test this out is the beta-1 adrenergic receptor, B1AR. So we expressed this protein in insect cells for simple post-translational modifications. We wanted, the easiest case. We mutated out some of the glycosylation sites. So we expect this protein to be relatively straightforward in being able to characterize it. So the green trace over here on the right is the simulated mass. So that's given the sequence that we expressed is what we'd expect the protein to be. And the black trace now is the measured native mass spectrum. And so you can see that these line up, they're identical. And so we can expect in this case that we have no post-translational modifications or nothing complex that would hinder this top-down analysis. So what we can do now is we can then select a peak of interest and fragment it. And so in this case, we've selected the +13 charge state. We can generate now a very, very rich fragmentation spectrum of, an abundance of ions. They're highly charged and actually quite large. And so I do just want to zoom in on this region here, just to show you just how information rich this mass spectrum is. And so we do see, a series of highly charged and again, very large fragments that we can then use to identify the protein. So if we take a look at what these highly charged fragments look like, we can actually start to pick out distributions that look like they would belong to one another. And they do in the sense that we're starting to lose sequential amino acids. And so we're generating these sequence tags now of components of this very large N-terminal fragment. At the end of it, we can generate these sequence tags. Same thing here for the +3 distribution. We generate a series of B ions with sequential losses of amino acids at the end of this N-terminal fragment. And then same thing goes for these Y ions at the C terminus. So we have a pretty large chunk of the C terminus and then these sequence ions that sequentially clip throughout the protein. And so in doing this, we can now combine, all the ions that we've identified. And so we can map it according to this map here. And virtually all of the fragments have been able to be identified in this, and that's because of these sequence tags. And so we were impressed with this. This is a 41 kilodalton GPCR, and we have 76% of the protein is covered. And so I'm not calling it sequence coverage. It's protein coverage. 76% of that protein has been, identified based on these fragments. And so we're excited about this because it gives us, not only, good coverage of the protein, but we do have these now sequence tags, and these do fall in the transmembrane regions. And so we're certainly not the first to see this. This is a well characterized for membrane proteins, and there has been extensive work done by Julian Whitledge and John Walker and the Kelleher group, and just really truly characterizing how membrane proteins fragment. And so we're seeing things that have been well characterized before. What's exciting for us, though, is being able to identify these things within the transmembrane domains gives us unambiguous sequence identification or protein identification. And we're actually quite excited about how much of this we actually see because denatured LCMS is one of the gold standards in being able to sequence these proteins, and native top-down. While it's a great technique, the sequence coverage just hasn't rivaled that of LCMS. And so this result tells us that we're getting closer in being able to truly characterize much more of the protein in native top-down. And so just to move on to just a little bit more complex of the system, this is the mu opioid receptor. I expressed this in mammalian cells, and so I didn't knock down any complex glycosylation pathways. This is just purely the native protein with the possibility for all native post-translational modifications. We do expect this protein to be constitutively active, so there could be phosphorylation sites, it could be signaling within cell culture, and so should be a relatively complex system. And so this now is the simulated spectrum in pink here. So again, based on the sequence that I expressed, I would expect it to have this mass and this distribution. But when we purify that protein and we run it by native mass spectrometry, we actually find something quite different. So this black trace is now the native mass spectrum. And we can see that it differs quite significantly from the sequence mass. So we do expect it to have quite a few post-translational modifications, that being glycosylation at the N terminus or phosphorylation at the C terminus, or some combination of both. We do, if we want to look at this a little bit closer, it's quite obvious that the peaks are split into two, but if we zoom in, we can actually see that we have several proteoforms that exist of this protein. And so what I thought was interesting is that the two major proteoforms. They differ by the sequence mass by 16 and almost 1700 Daltons. But these two major proteoforms only differ from one another by 162 Daltons. And so this tells me that, these proteoforms could differ by a single Manos even. And so we can, speculate all day using the intact mass. But if we truly want to characterize this, we're going to need to sequence it using native top-down. And so we can then select our +14 precursor, and we can generate, again, a very rich fragmentation spectrum full of highly charged and large ions. We can start to do the same type of analysis where we can look at a series that looks like it would belong to one another. And we do, again, start to see some of these sequential losses of amino acids. And when we put that onto the map, it is, again, within the transmembrane domains. And so we can start to parse out certain parts of the protein. But this becomes a little bit more challenging because with top-down fragmentation, we typically get fragmentation of the termini. And in the case of GPCRs, both termini are heavily modified. And so at the N terminus, we have 5 putative glycosylation sites. And at least one of them is certainly occupied. And then at the C terminus, we have at least 6 phosphorylation sites. And we do find that, again, at least one of those is identified, but not yet localized. And so this is an ongoing effort in being able to use top-down fragmentation to target these very complex membrane proteins. And so we will have to use some additional fragmentation methods, maybe electron-based fragmentation methods to really get at these cytoplasmic and extracellular bits. But the idea is that having these sequence tags in the transmembrane domains is very exciting because while we won't need full sequence coverage, we can still unambiguously assign these fragments to a specific GPCR. And so the real motivation and the major motivation behind this is, for our newest endeavor. And so we were recently funded under the Welcome Leap program. It's called Multi-Channel Psych, and it's a series of teams that are working to decipher the molecular mechanisms of anhedonic depression. And so what we're doing now is we're taking these endogenous receptors and transporters and GPCRs, and we're extracting them from human brain tissue. And we're using native top-down mass spectrometry to be able to parse out the complete molecular ID of all of these interacting components. And we'll do this for unaffected patients, but also depressed patients, to be able to better understand how these receptors and how the different proteoforms are influencing, how our brains work and how we process reward and motivation and function. And so this is just a picture of me just this morning. I do recognize that many of you that are calling in from the States are either eating lunch or having coffee, so I apologize. But this is me dissecting, not human brains in this case, this is very much of animal origin. But we're extracting microvessels from brains to be able to do this. And so these developments that we're working on with Thermo and being able to fragment these GPCRs in this way is huge because endogenous proteins are quite limited. We have a finite amount. And so being able to do this type of analysis in a very robust way on all-in-one platform is going to be huge for making an impact in depression treatment. And so just to conclude, the developments that we've made with this IR laser gives us now tunable parameters in terms of laser power and irradiation time. So we can now well control how the membrane protein gets released from the detergent. And we found it's extremely advantageous for the subsequent top-down analyses that we're doing. Having the iron activation prior to top-down has given us 74% coverage of just a test system, which is B1AR, but it has proven itself to be a robust means for sequencing and being able to identify more complex proteoforms. And so this work is still ongoing. In the Orbitrap Eclipse, we do have other means to fragment. And so we're going to start incorporating some electron-based techniques to find some complementarity in the coverage. And so starting to get at, those extracellular domains and the cytoplasmic bits to characterize the entire protein all in one go. And then, of course, we're still optimizing these tunable conditions to preserve these more fragile interactions. And of course, we're now applying these workflows to these endogenous proteins that we're taking from human brain. And, these proteoforms are complex and abundant, and it's very expected that, from a living system that we're going to see a lot of variability. And so with that, I want to thank Carol, she's the director of the Kavli Institute and the boss behind all of this, and Tarek is another co-PI on this grant with us. John, Josh, Chris, and Ian at Thermo, Eddie at OMass, and support from the guys at Proteinaceous with a lot of this top-down analysis. In particular, Ken Durbin has been a huge help. The funding that made this possible, and of course, all of you for tuning in tonight. I'm happy to take any questions you all have.
Great, thank you, Corinne. Excellent work. Totally fascinating. Can't wait to see where this all goes. A few questions coming in here. We have, where'd it go? Joe Liu has a question about what is the detergent you use for the native MS, the membrane proteins, and is it protein dependent?
Yeah, so we use a lot of DDM is our go-to. It's a pretty good universal detergent for a lot of membrane proteins, but not all membrane proteins like the same thing. So we do several detergent screens. Art Laganowski's paper from our group outlines quite a few that you can use. There's also a detergent that we made in-house. We call it G1, but there's a publication about that as well out of Carol's group. But I guess the short answer is that it is protein dependent, so it's something that we have to optimize.
Thanks. Moe had a question. Hi, Corinne, very exciting talk. For your human animal tissue work, are you using antibody-based purification or some physical property-based methods?
Antibodies, yeah. Immunoprecipitation.
Okay, great, thanks. Neil had a question. Great examples of proteoform measurement in biology here. Can you say more about what is known on glycoproteoforms modulating agonist-antagonist binding to GPCRs? Cheers and great talk.
Yeah, thank you. So there's been reports of, the one that comes to mind are chemokine receptors. Those GPCRs have, I think, about 3 glycosylation sites normally on the N-terminus and the extracellular loops. And there have been several studies, not by me personally, there's a lot of literature out there that it's not just the presence of the glycosylation, so whether site is occupied or not, but the types of sugars. So if you have sialic acids on some of these sugars, you can completely abolish binding of certain cytokines. And so there's a lot of implications in, sugar disorders and, the different, sugar biology that each person has and how their receptors would behave. Given the same ligand, some people might not be able to bind those appropriately.
Thanks. And Joe had another question. For elucidating the sites of PTMs, have you considered going the denatured route like the Whitledge method using chloroform formic to get higher charging, which should help the top-down MS work? Yeah,
and so so we will. We have been doing that because there's truly no better. LCMS is the gold standard, right? And so there is complementarity in the methods where we're using the native top-down because the native allows us to directly relate the ligand to the proteoform of interest. And so it's a way to kind of unify the ohms. But then we still do need to do these LC-MS denaturing and proteomics type techniques to really fully sequence the PTMs and the entire protein.
So I had a question, more of a general question. Also, if folks want to ask a question live, they can just unmute themselves or ask to be unmuted, and they can go ahead and ask. You mentioned sequence coverage is somewhat a limitation currently, and you mentioned a couple areas about other activation methods and things like that you can employ. What do you think will have the biggest effect in extending sequence coverage going forward?
Oh, I think for us, it's just a matter of, imparting enough energy to essentially unfold, right? So there's a lot of work on, how the tertiary structure dictates how something fragments. And so I think, on a protein by protein basis, especially if we're doing this in the native state, there's a lot of tertiary structure effects that we're having. And so my go-to is always more energy, which is why I like the laser. But yeah, I think higher signal, more energy.
So there's a message for anyone building instruments out there as to how to get there. So what about going forward? You mentioned your new project. I mean, where do you think, like in 5 years, assuming that all this technology really evolves and you can get so we can routinely do these things, what kind of impacts do you see this being employed routinely, this kind of measurement to determine ligand binding or effects like this? Or where do you kind of see it going ultimately?
Yeah, I mean, well, I guess you're asking, diehard mass spectrometrists, and I firmly believe that mass spectrometry can certainly answer a lot of these questions, and I'd love to see it like this in the clinic even. But I do think, we are getting closer, if we're not already there, even with the Proteoform project, like truly understanding the vast diversity of the proteins that are out there. And I do think being able to connect proteoforms to their ligands, to the oms, we can start to fill in the missing link that is, from genotype to phenotype, molecules are the center unifier. And so, yeah, I think mass spec has absolutely the #1 spot in being able to decipher that when it comes to health and disease.
Great. And a question from Jake. Have you considered any native separations prior to the MS for the endogenous tissue work?
Yeah, so we haven't employed it yet, but it is something that we will have to explore, because I think, the number of proteins that come out of these endogenous tissues is enormous. And so there will certainly have to be, with the immunoprecipitations that we do, additional steps for separations.
Great, thanks. Are there any other last questions? New questions, new ideas, concepts? If not, then I'd like to thank Corinne for fascinating work. I can't wait to see where this all goes as it evolves. This really great stuff, this whole arena of native and ligands and binding, etc. So thank you very much. We really appreciate that. I'd like to thank everyone for attending in this busy time of the year, and wish everyone all the best for the holidays, and we'll talk to you next year. So thank you again, Corinne, and goodbye, everyone.
Thank you.